Hello, everyone, and welcome to today's live broadcast, Controlling RNA-Seq Experiments Using Spike-in RNA Variants, presented by Dr. Lukas Paul. We're excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by Lexogen. Lexogen is a transcriptomics and next-generation sequencing company focusing on development of products enabling complete transcriptome sequencing. It was founded in 2007. Lexogen is based in Vienna, Austria, and has a subsidiary in New Hampshire, USA. To learn more, please visit www.lexogen.com. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located on the lower left of the presentation window and type your question into that box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. If you'd like to enlarge that window, just click on the screen icon located at the lower right. Now, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or just use the Q&A button to let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Now I would like to introduce Dr. Lukas Paul. Lukas Paul, PhD, is Head of Services at Lexogen. Dr. Paul received his PhD in Biomolecular Sciences from the University of Manchester, UK, for work on mRNA stability and translation. He continued as a postdoc at the University of Vienna and as a staff scientist at the Max Parris Laboratories, focusing on RNA folding, RNA-protein interactions, and fluorescence assay development. In 2008, Dr. Paul joined Lexogen as a research scientist, developing RNA-Seq protocols. He is now Head of Services, managing sequencing projects for Lexogen's customers, and also developing standards for RNA-Seq applications. Please join me in welcoming Dr. Lukas Paul. I will now turn the presentation over to him. Dr. Paul?
So, thanks a lot, Christina, for this kind introduction, and welcome to you all. So this webinar today will be about the use of spike-in RNA variants to control RNA-Seq experiments. And I will start straight away with still a short introduction to Lexogen. Not all of you might know this company. We were founded in 2007. We are headquartered in Vienna, Austria, where we also have our production and R&D facilities. And actually, a third of our staff is still in R&D, and we are focusing 100% on RNA sequencing tools. So that means that Lexogen is really focusing on transcriptomics and on RNA sequencing. Now, depicturing the RNA sequencing workflow to the right, you see that you usually would start with your cells or your tissues. You would extract the RNA and convert it to cDNA, which is then wrapped up in NGS libraries for short-read or long-read platforms. And after sequencing, you then have to evaluate your data and interpret it. Now, at Lexogen, we have developed a lot of kits for all kinds of applications along this workflow, starting from the RNA extraction to library preparation up to bioinformatics solutions. But I will not be talking about these today. If you're interested in them, you're welcome to visit lexogen.com. But today I will be talking about something that encompasses the whole workflow, and these are the spike-in controls. So this webinar will be structured into first talking about why we need valid controls in RNA sequencing. Then we'll talk about SIRV design structures and features. We then will see how the SIRVs are produced and mixed, and how the spiking of the samples is best performed. And this will already bring me to our SIRV suite, which is a Galaxy-based collection of tools, which has all modules necessary for the whole spike-in experiment. And I will then finish this webinar with an outlook on future spike-in modules, And in the lower part of this slide, you already see a few of the links that might be relevant to you. Most importantly, at lexogen.com/SIRVs, you have all the links and all the information that you need if you're interested in the splicing control. So why are valid controls necessary in RNA sequencing? If you look at a standard RNA experiment, like the separation of RNAs on an agar gel or using a microfluidics device, you would say, yes, I need splicing controls. And this would then take the form of a marker or a ladder so that you can size actually your RNA product, your RNA band, and you can also quantify it. Now, this is the point where this webinar becomes interactive. So it's the first time we do this, and I will push out the poll. And that should be working right now. So I want to ask you, the audience, have you used RNA spike-ins in NGS experiments before? So you can answer yes or no, and I urge you to do it pretty fast because I think in 5 seconds this poll is already closing. That's pretty fast. So, wow, I have to say half of you participated. It's really good. And, well, it's almost leveled out. So 22 of you said, yes, I've used RNA spike-ins in NGS experiments before, and 33 no. So it's a close call, and actually that's also what we have seen before. So 2 years ago, there was a similar poll at the blog platform RNA-Seq blog, and also there half of the people said, yes, I've used them before. And that is very encouraging, but still we think that more people should use splicing controls, because if you think about the efforts that are involved in an RNA sequencing experiment, you should really include the controls to be able to evaluate not only your experiment, but also to compare it later on. That's the reason why you should use controls. So the use of the controls makes the samples comparable in one experiment and also between experiments. So only when using external controls, you can know your technical variability between the samples and the experiments. And only once you know this technical variability, you can then start investigating the RNA biology or the biological variants behind that. So, and this technical variation actually is introduced at all stages of the workflow. So from RNA extraction, which is actually a very variable step, to NGS library preparation, sequencing, and data evaluation, we have all kinds of factors that introduce variability, be it the operator, the purification kit, the library preparation kit, the read node, single and paired end, chemistry, version 1, 2, 3, 4, up to the bioinformatics tools. You might think, at least the bioinformatic tools, that should be a stable configuration. I've set it up and now I'm just using that one pipeline. But you shouldn't forget that this one pipeline is only one variation that you're using. And looking up at Wikipedia, for example, you see that you've got all kinds of tools for all steps along the pathway. So you've got a large number of tools for trimming, bias correction. You can choose from any of those splice aligners available. And the largest variety is at the level of normalization and quantification. Now, if you just multiply all these possibilities, you get to a very high number of variations that are possible. And even if you do not explore all of these variations, just think about the possibility that you now actually evaluate your transcript with, let's say, top hat, and then in 1 year you change to top hat 2, and that's a version upgrade that affects the quantification, and you should be able to control for it. So external controls can now measure this technical variability. And the step where you add the controls is actually at the sample step, so either at the RNA extraction step or in the cell homogenate, and you only have to add a very tiny fraction of these controls, meaning that we usually would recommend that about 1% of your final reads should map to the controls, and therefore you do not have to sacrifice a lot of your sequencing depth to these. These controls now are treated the same as the endogenous RNA, and that means that actually the characteristics are tightly linked. However, at the level of the alignment, you can now separate the controls, so the reads mapping to the controls, and these can then compare. You can do an input-output comparison. And based on this, you can now determine the accuracy of your experiment. And if you have replicates, which is highly recommended, then you can also determine the precision. Now, once you've got these quality metrics values, you can determine whether actually you should compare those samples and those experiments that you've spiked in, because only the characteristics of this small number of reads mapping to the spiking controls informs you about the endogenous RNA and the experiment itself. Okay, so now we have a bit of a time warp here. So we go back to the year 2003, where the first spike-ins have been introduced. And these were developed actually by the External RNA Control Consortium, the ERCC, which was hosted by the National Institute of Standards and Technology. So they devised actually controls for microarrays, and they produced 92 transcripts from 92 different genes. So these are non-isoform single exon transcripts. They have viral sequences, and they mimic human mRNAs to a large extent, meaning that they've got a similar size range and GC content, and they also have a poly-A tail. Now, they've been commercially available for quite some time, so there's a large number of publications from the 2005 original publication up to the ERCC dashboard from Sarah Munro at NIST. One read that I could recommend is the Nature Biotech issue from September 2014. There, the SEQC consortium actually evaluated many different RNA sequencing platforms and workflows, and they also evaluated the ERCCs along that. So this their initial generation of spike-ins, what can you use them for? You can use them to detect your dynamic range and your limit of detection. You can assess fold change, dose response, and many other technical assessments. I will show here only now the dynamic range assessment. So here you see that in this particular experiment we've used one of our kits, which is called QuantSeq, and this QuantSeq kit derives only one read from the 3' end of a transcript, meaning that you just get one fragment, and this 50 base pair read is actually already enough to identify the ERCCs correctly. And all 92 ERCCs can be identified correctly and also quantified more or less correctly. You see that the input-output correlation clusters along a straight line. So that is a very useful device and tool to assess your limit of detection and the dynamic range. However, these ERCCs are non-isoform controls. And because they have no transcript variants, one of the major challenges of sequencing, and that is to assess complex transcriptomes and to identify and quantify splice variants, this cannot be evaluated. And that was actually the reason why we set out to develop external controls that can address exactly that, alternative transcription and splicing. So in 2013, we designed the SIRVs, which were then presented in July 2014 to the ERCC 2.0 workshop. This is actually an initiative promoted and fostered by NIST. And later on in the next year, we had a test program where the SIRVs were evaluated on multiple platforms and at multiple sites. And we could then release these SIRVs to the market, and the commercial launch was last year in September. So now that we've introduced the concept of the SIRVs, we want to look at them in detail. So the SIRVs are actually modeled on human gene templates, meaning that we took sufficiently complex human genes, like, for example, KLK5, and we also downloaded the annotated transcripts from the Ensemble database, looked at it if it covers all the splicing and the transcription complexity required, and we then also added some transcripts. For example, here you see that we've added overlapping transcripts, both in the sense and the antisense direction, but we also added some minor spliced variations, start set and end set variations as well. And you also see a couple of transcripts that are actually depicted in green. These are not realized transcripts, meaning they are not present in the RNA mixes as RNA molecules, but they are a part of an over-annotation. So this means we not only have the correct annotation available, but we also have an over-annotation where more transcripts are annotated than actually are present in the mix. And that's actually a real-life example where not all of the transcript variants are expressed in the given tissue, and your whole workflow, your bioinformatics pipeline has to cope with that. Also, vice versa, we have we have one annotation version, the insufficient annotation, where we remove some of the transcripts from the annotation that are actually present in the mix. So that would be the situation where you have to deal with a not well annotated transcriptome, or where actually in your sample there is a new transcript variant that has not been annotated before and that you have to deal with anyway. So we not only have one gene, of course, but we have 7 of these. And all 7 of these actually have a very high complexity. So this means the SIRVs mimic the transcriptome complexity in a nutshell. And we want to test actually the complexity to its limit and not have very simple case examples with only 2 or 3 transcript isoforms per gene, but this should be really a challenging setup. So in the end, we obtained 7 artificial genes with 6 to 18 transcript variants each, summing up to 69 in vitro transcript variants. The length mirrors again the nature of the human mRNAs from 200 to 2,500 nucleotides. Also, the GC content is in that region. All of these have a uniform poly-A tail, which is 30 nucleotides long, and guarantees in standard library preparation protocols that these SIRV mRNAs are captured with the same or almost the same efficiency as endogenous RNA. We also observed the exon-intron junction rules, meaning that most of them have the canonical GT-AG dinucleotides. However, we also have included a minor fraction of known exceptions. The intron sequences are also defined, meaning that the annotation has intron sequences and are not left blank. These are random and do not align to known genomes and transcriptomes, and actually also maintain the sequence properties of the neighboring exons. Now, the exons themselves actually have been tested thoroughly. So the sequences for these exons are natural sequences, and we inverse them, meaning that we flip them 5' to 3', and that retained the nucleotide composition, but the sequences completely lost their original identity. And we confirmed that by blasting our SIRV-Home, so the entirety of the SIRVs, against the NCBI database, so against all sequenced genomes and transcriptomes, and also against the ERCC sequences, and we found no significant similarity. We then also proceeded to check that in an experiment. So we had one RNA-Sequencing experiment where we had only the SIRV transcripts present in the library, and we saw that of all those close to 10 million reads, they exclusively mapped to the SIRV annotation and not significantly to the human annotation or other model organism annotation or the ERCCs. So, in fact, we've shown here that you can combine the SIRVs with any of your endogenous RNAs from different sources, being it yeast, being it Drosophila, or human samples, and you can also combine them with the ERCCs if you want to. So that is now a global view of the SIRVs. We like to think of this as a SIRV-Home. You've got here, like pearls on a string, the SIRV genes with all these 69 transcript variants. Our SIRV gene definition also includes 1,000 nucleotides upstream and downstream, so these are defined sequences. And taken together with the intron definition, we really have now the full SIRV gene defined, which is important for some of the bioinformatics applications. All the sequences are, of course, freely accessible either at our homepage, lexogen.com/SIRV, or they have been also deposited at GeneBank. Now, talking about spiking isoforms, I also want to mention the sequence. So these have been developed actually by the group of Tim Mercer and colleagues at the Garvan Institute. They were published just like 2 months ago, and they have a similar concept to the SIRVs, meaning that they constitute a transcript variants of a given gene. And actually, there are quite a number of genes, so 78 genes encoding for 164 alternative isoforms. But that also means that actually that transcript number per gene ranges from 1 to 4 for the segments. So on average, there are only 2 transcripts per gene, which puts them a little bit closer to the ERCCs, whereas the SIRVs has a very high complexity with 6 to 18 transcripts per gene, and on average, we have actually 10 transcripts per gene. However, the transcript number per gene is not the only definition of complexity. It is also important how many overlaps and similarities of the isoforms you have. And as I mentioned before, we've put a strong emphasis on making this as complex as possible to test the boundary conditions. And complexity in a nutshell also means to us that we want to reduce the number of control genes. So now that we have defined the SIRVs, we want to mix and spike them. But first we have to produce them. We produce them as T7 transcripts. So you see here actually a gene cassette that's cloned into a vector, and of course it's all sequenced and sequence verified. Because of the T7 transcription, all our RNAs have a triphosphate at the 5' end, and we have hard encoded 30 nucleotides at the poly A tail. And we also produce quite long RNAs, so 14 of these SIRVs are actually longer than 2 kb. This is actually harder to produce, but on the other hand, the short RNAs are sometimes the ones that are the hardest to determine in an NGS analysis. Still, we have to get all of these RNAs, all of the 69 RNAs, at a very, very high purity and foremost integrity. So we do not want to have any degraded RNA or any byproducts from the transcription. And if the transcription itself wasn't very pure, like in the left-hand case for SIRV 612, we applied custom purification methods to really have a very high integrity and no degradation product. Because the degradation product of one isoform can then later on heavily influence the quantification and identification of all the other isoforms in a given gene. We follow this high quality through the whole mixing scheme. So a number of SIRVs are combined in premixes where we can still determine exactly the number and the quality of the SIRVs that are in these premixes. We tightly control how then they are combined to submixes. So we have now 4 submixes. These, of course, have high quality values. And these 4 submixes we then combine into 3 final mixes. The mix E0, E1, and E2. You already see that in the mix E0, we have all the 4 submixes at the same ratio, meaning also that all the SIRV transcripts are present at equivalent concentrations and at the same level, whereas in E1 and in E2, they are present at different concentrations. Now this can be nicely illustrated in that diagram here. So you see that at the 0, we've got all the submixes and all the SIRVs at equal levels. In mix E1, we have now the different variants of a given gene in the different submixes up to 8-fold different concentrations, and in E2 actually this concentration goes up to 128-fold. Still, please keep in mind that actually sometimes also here it is more tricky to actually detect 1.5-fold or 2-fold difference than a large 128-fold difference, which is sometimes rather a function of read depth and sensitivity. And of course, you can also determine now the ratios of this mix. So imagine that you have now 2 samples. One sample is spiked with mix E0 and the other one with mix E1. You can now determine actually the differential gene expression on the transcript level, and that is shown here. So the SIRV product itself contains now all these 3 mixes, E0, E1, and E2. you will get 4 microliters, and you can draw, let's say, 1 microliter or 0.5 microliter. And this can then be diluted up to 1,000-fold, and that provides now spiking RNAs for thousands of samples. We have to deliver that at this high concentration, at about 25 nanograms per microliter, to maintain the integrity and the stability of the product. And of course, this is RNA, so when you receive it, please be careful, store it at -20, better at -80, and apply all the good laboratory practice rules that you know. Now that we have created our mixes, we can perform a SIRV experiment. And this is actually now the occasion where I can introduce the SIRV suite to you. The SIRV suite itself is a set of software tools that is freely available. Actually, we've got a cloud-based instance running this SIRV suite. And it is created in the Galaxy environment. So many of you might be accustomed to that environment. It is accessible at sirvsuite.org, and actually it is structured into major modules, and you would start your experiment with the designer module that aids in planning the experiment. You then can perform the experiment, you upload your data, and then you enter the metadata for your experiment. That is done in the specifier. The core module actually is the Evaluator. So the Evaluator can evaluate your SIRV data and obtain a standardized report. And you will also get all the data files that are necessary for the publication. This is a PDF report, but you will also get all the vector graphics and all the raw data. And this data is also then used actually in the comparator. So the comparator actually performs now the cross-sample comparison. So meaning that what has been produced in the Evaluator can be stored in the database, and the comparator now takes this one and can tell you if 2 samples, 2 experiments have a high concordance and should be compared with each other or not. So that's a screenshot of the Galaxy SIRV suite. It can be used without registration. However, we recommend registration because it's pretty much straightforward. You then can save data and compare against other data sets. Of course, we respect the data privacy, meaning that only the SIRV-related data is stored for comparison. And the SIRV suite is actually offered in a cloud instance, free of charge. However, you're also free to clone this SIRV suite to other cloud-managed instances. And the SIRV suite can, of course, already also be installed into existing local Galaxy installation. And one of the major driving forces behind this creation of the SIRV suite was that we realized that the data analysis is always the biggest challenge in any RNA-Seq experiment, and even more so with the controls. And by implementing all these tools and also predefining a pathway, we want to realize one way how you can analyze the SIRVs. Of course, please feel free to come up with many creative ideas how you can use this control, and this is just one realization. So those of you that are accustomed to Galaxy will know how it looks like. You've got access to all the main tools and also some helper applications. And of course, we will start with the experiment designer. Now, the experiment designer actually is giving you information for the step where you spike in your SIRVs into your RNA, your endogenous RNA. It will look like this. So unfortunately, I just have a screenshot here. However, you have to imagine that you can now change all those values. Actually, all those bars here are scroll bars. You can just drag and drop them, or you can enter values directly. So in this example here, we would start with 10 nanograms of total RNA. And because we say this is a human brain reference RNA, we know the mRNA content, which is 2% here. If you do not know the mRNA content, you can speculate on that or leave it out. What you will get is the mRNA in your sample, but you can also choose to say, okay, I have already poly-A selected mRNA, then you can enter the value directly here in the fourth column. Or you say, no, I've done a ribodepletion experiment, and then you can also determine the ribodepletion efficiency. And in the end, what you will get is the output in the middle. So the purple gradient in the middle in the concentration range window shows you actually now where the reads from the endogenous RNA will be distributed. So you'll get an estimation of the number of molecules and the concentrations where your endogenous RNA, which your endogenous RNA will cover, and also the TPM values. And the same is true for the controls. So on the right-hand side, you now see that we have here preset 1% concentration for E0, for example, and E0, E1, and E2 would then actually determine at this level that you get a concentration, that is up here. So meaning that you will get quite high TPM values for your SIRVs, and that means that you get enough reads to cover your transcript isoforms, and your pipeline should be able to identify and to quantify your isoforms to a very good degree. We also have already included the option to calculate the same for the ERCCs, so we could also include it at that stage here. So the concentration and dynamic range covered can also be depicted in a classical input-output correlation diagram. This one here is actually taken from the user guide, and that's also true for the next slide. So that's the user guide of the SIRVs actually. Here we have a run-through of a typical experiment. In that particular case, we were aiming at 3% spiking level in the final reads, and we were starting with 100 nanogram of RNA. Now, there were two options to perform this experiment. One is either with a poly-A selection, and the other one with a ribodepletion. And in both cases, here you see what is the volume of the SIRVs that you have to spike in. Please note that 1 to 1,000 dilution, so that a SIRV sample is sufficient for a large number of samples. And this particular realization has been performed for a reference RNA set. So we have created reference RNA samples, RC0, RC1, and RC2. They are modeled on the FDA's MAQC reference RNA samples, set A and set B. So in RC0, you've got the universal human reference RNA spiked in with the SIRV mix E0 and also with the ESC export mix 1. In RC1, you've got the human brain reference RNA spiked with SIRV mix E1 and ESC export mix 2. And RC2 has a mixture of the reference RNAs and is spiked with the SIRV mix E2. So this is also evaluated in our manuscript that we have deposited at bioRxiv. You see a link in the bottom part of the slide, or you can also always access everything via our homepage. So now we get to the evaluation. Now that we have actually mixed our RNA sample with the spike-ins, we want to evaluate the data. So we want to evaluate how library preparation and sequencing affected our spike-ins and our endogenous RNA. And this means we first have to get the data into the SIRV suite. And you can either upload your data from files that you have created, or you can also access it in the SIRV suite directly. The reference experiment data is directly accessible in the SIRV suite, or you can also download it from the short read archive. It's all up there and accessible. And then you have to enter the metadata for this particular experiment. So this is pretty much self-explanatory. You have to give the experiment a name, which library prep you used, and so on. You will get all kinds of help, either directly in the form on the bottom of the page, and there's also actually a user guide available for the SIRV suite and the help file. So now that we have uploaded our data and specified the metadata, we can now evaluate our SIRV reads. And this is done now with the SIRV experiment Evaluator. So the Evaluator actually calculates the ratio between expected and measured reads. And it does that actually in relation to the endogenous RNA. And that means that if you have, for example, used 100 nanogram of total RNA input, and you're sure that you have spiked in 1% of the SIRVs, then the evaluator can calculate the mRNA content, so the percentage of the reads that map to the endogenous RNA will give away now the mRNA content. But more importantly, the evaluator can now examine the experimental variability, and to do this, the evaluator actually generates a compact coverage visualization graph, which I will explain in the next slide. And this coverage visualization graph is then used also to derive a single figure for the deviation between the measured and the expected coverage, and that is the coefficient of deviation. So that's already the first value in our quality matrix. Of course, you will also get a list of telling junction reads and contrast that with the expected number of telling junction reads. And the second quality measure that we can derive from the SIRVs, but also from the endogenous RNA, is precision. So precision actually measures the scatter of calculated abundance values, and it is the mean of all SIRV standard deviations. So as long as you get the replicates, you can calculate this precision. You could also calculate it from the endogenous RNA. The third quality measure that we can derive is accuracy. So accuracy measures the deviation of the calculated abundance values from the expected value, which means that here it measures actually the deviation of the measured versus the relative SIRV concentration, and that's the median of all absolute log two-fold changes. And it's important to note that accuracy can only be measured if you use controls. You have to know the input. The fourth measure that the evaluator actually can derive is differential accuracy if you have actually done a differential gene expression experiment. So here again you see the pipeline of the evaluator. It actually processes experimental data from SIRV controls and endogenous RNA in several different stages. You also see the white arrows here. I'll try to mark them. E1, so that would be actually your read data that gets into the pipeline. You could also already add trimmed data. You can enter the pipeline by having already mapped reads, and actually that's the stage that is realized at the moment in the SIRV suite. So at the moment you have to use already mapped data to enter the evaluation pipeline, but of course first of all the other modules will be added in the near future, and secondly, because this is a Galaxy environment, all those mapping tools like STAR, trumpet and so on are available to you. Then actually within the evaluator module, you can then assemble the transcripts and quantify them, for example, with cufflinks. And it is important to note actually that we have not only one annotation for the SIRVs, but three, so the complete, the correct one, an insufficient, and then over-annotated one. So if you want to really challenge your pipeline, you can use those template annotations, or you can also create your own ones. So now that we have actually quantified our transcripts, we have to do a normalization step. And this normalization step I will explain on the next slide, whereas the output of the evaluator is a report and of course all the data files required and later on to compare experiments. So the normalization actually includes that we identify outliers, and these are identified in trumpet plots. So the basis for the prominent outlier, the TPM artifact identifications, are these trumpet plots of technical replicates. It could be, for example, that in one replicate a short transcript would receive no read, so 0 TPM, and in the other one a fraction of a read. And because it is such a short annotated transcript, actually one of the TPM values would be really high, and this filtering identifies these outliers and adjusts it to a threshold that you have defined before. Also reads below the threshold or set to this threshold, and yeah, that's what's done here. And now we actually get to the first result. And to introduce you to the first result, I will show you a very familiar picture. So this is something that you encounter in most product flyers. That is the accumulated coverage of the top 500 or 1,000 genes. This is more or less informative in terms of determining the overall bias, and if you have heavy RNA degradation, you will see that. But it's actually of no use to look at individual transcripts and if the data sets are actually comparable, if you could compare the data sets shown here. To do this, actually, you have to go down to the gene level. And depicted here is, for example, SIRV-3. Because we know exactly the input and the contribution of all the transcript variants, we can calculate the expected coverage, and that is shown in blue for the sense direction and in red for the antisense direction. The view that you enjoy here actually is the condensed coverage view. So here we have normalized the length of the introns because we feel that actually that is the only way how you can have a meaningful overview of the coverage. And you also see that we have quantified the junctions. So you see the normalized junction coverages here, and this is contrasted with the expected junctions. And you see that in this particular experiment, the coverage is sometimes spot on, and sometimes it's too low or too high. So that's what you would usually expect, and what is your pipeline then actually also correcting for. It's very interesting to actually compare these coverages. So this particular two coverages were derived from sequencing the same RNA at two different locations on similar but different platforms. So you see actually here the expected coverage in blue that accounts already for the edge effect, so that you have not such a high mapping to the 5' and 3' end of the transcripts. And you see that in many cases, actually, the coverage derived is exactly the one that you would expect. And in many other cases, that both or one of the coverages are very different from the expected ones. It's also nicely seen that you see the coverage contribution of one transcript here. So that is actually one exon contributed by only one variant. And you also see that in terms of junction coverage, you sometimes have a high agreement and sometimes you don't. So already here, also it's the same RNA, but sequenced on at different locations, there's a high divergence. Now it would be actually nice to not always have to overlay these coverages and do this for all the genes, even if it's only for the 7 SIRV genes, but to derive a single value describing this coverage. And that's what the coefficient of deviation does. So the coefficient of deviation is actually a value for the coverage target performance comparison. So it is derived by calculating the average of deviation at each position. And this is maybe best illustrated by examples. So the ones that I've shown you before is actually not too bad. So the coefficient of deviation is 0.23. So if you got a low COD, that's actually really good. And then you've got the even coverage, and the bioinformatics pipelines will probably work fine. If you got already a bias in your sequence and an uneven coverage like shown here, then this will be reflected in the COD value. It will go up here to 1.66. And actually, just by comparing the COD values, you would already think, okay, I don't know if I really can compare this to experiments. I've included also one extreme case where the RNA was degraded. So that gives you a COD value already of more than 5. However, that could also be because you are using a 3' mRNA sequencing method. Nevertheless, that's already a first quality measure that is important to get to decide whether the samples are concordant or not. All of this is actually then published in a PDF file in a report, which looks like this. However, we have also other results from the Evaluator, and one of these other results is a box plot-based comparison of the measured and the expected SIRV concentrations and ratios. So depicted here are now the SIRV transcript concentration distributions within the 4 submixes, and they are now correlated with the measured abundances. So within this box plot, you see upper and lower quantiles being represented by short bars, and the individual transcript abundances are gray dots. You see already in E0 that most of the SIRVs are not quantified correctly, although actually they should be present at equimolar levels, and that's also true for E1 and E2, which also have extreme outliers. So it would be now interesting to see actually whether the quantification of these individual SIRVs has the same bias in different mixes. And we'll try to look at this particular case here. So you see a scattering of those quantifications in E0 and E1. However, if you look at the differential gene expression, you see that that bias is actually accounted for. Okay. So this input-output correlation plot is also one alternative to the box plot, and you see that actually the ranges where E0, E1, and E2 are present correlates well to the expected values. The SIRV suite report also includes this kind of correlation plot here shown for another experiment, and it also calculates R-square and p-values in both the linear and the log-log space. And now we actually get to a very interesting input-output correlation, and that is on the transcript level. So in this heat map here, actually, you see the testing of 2 different algorithms. So again, the same RNA dataset, the same RNA sequencing dataset was quantified by 2 different algorithms, D1 and D2. And the Evaluator actually calculates the accuracies for all individual transcripts as log-2-fold change of the expected to the measured abundance. So shown in green are here now log-2-fold changes between -1 and +1. That corresponds to a 2-fold lower or higher concentration. In red, you've got all those values with more than 1 log-2-fold change, and in blue, the ones below -1. Now let's look at a few examples. For example, here, SIRV-613. So the first pipeline, D1, actually is doing quite a decent job. It doesn't have a high deviation from the expected value, and the same is true for D2. So also here, actually, they show quite a good concordance. The same is actually true for SIRV-611. Also, actually, both of the pipelines overestimate the abundance of all of these transcripts. But because the bias is the same, actually, we still show a very high concordance. Of course, there are also other examples where this is not the case, like here for 617. So one pipeline is over the other, underestimating these abundances, and here the concordance is really, really low. So, having now evaluated our SIRV experiments, we can store all this metadata and compare it. And this is done in the comparator. You see that you get all the data from the evaluation pipeline, and you can now actually choose to compare 2 experiments with each other, or you can also compare it against the experiments stored in the database. And only the SIRV-related reads and the SIRV-related data is actually stored in the database and is needed for this particular application. That's the interface. And with this, I get to the definition of concordance. So this can only be calculated on the SIRV controls, actually, and not on the endogenous RNA. It is actually independent of the accuracy of the individual samples and experiments, but it describes the coherence of this data set. So what you actually do is you compare the quality matrices, and the concordance is actually the median of all logfold changes, the absolute logfold changes calculated for the SIRVs in 2 experiments. So essentially, it is the relative accuracy measure calculated by comparing 2 experiments with each other. And a high concordance is represented by small values, actually. So if you would imagine not having 2 but 4 algorithms and software pipelines, and they all have different accuracies, you can then calculate actually the concordance. And in that particular case, for example, examining the data set with algorithm 1 and algorithm 3 would be a good idea because of the low concordance value. If you would choose to compare one data set with algorithm D2 and the other one with D4, that would not be a good idea in that case. So what do you really get from this concordance calculation? As you've seen, you can assess how a change in algorithm actually affects your data evaluation. You can monitor the stability of your data evaluation across samples, across experiments, across also sites with your collaborator, and also over time. You do an experiment now or in 6 months' time, and you can compare how stable actually that is. And a very nice feature is actually that you can derive a very good estimate about the false positives and the true positives, because the concordance already gives you the background difference between experiments, and from this, for example, you then can calculate how many validation experiments are necessary, how many QP cells you have to run, and so on. And maybe most importantly, you can use these concordance values to find data, for example, at the SIRV-Suite database that have also the same high concordance, and you can then contact or download the other data and do a meaningful correlation here. So with this, I almost are at the end of this presentation. I want to have a quick outlook on future modules. And it's actually important to note that the modules here are actually here to stay. So they will remain and they will be compatible with the other modules, like the SIRVs are compatible with the ERCCs. There will be more and additional modules to follow. We sort of have to get to an internal ranking, of course, but we try to keep this modular nature. And that might be illustrated by the kind of modules that we can think of, so that there's no particular order, but you could think of spiking transcripts that are longer than 2.5 kb, of course, up to 10 kb, 20 kb. That's important for long-read sequencing. You could vary the poly A tail length. You could have nucleotide modifications in the poly A tail, of course. You could think of controls without a poly A tail. Chimeric RNA spike-ins could test transplicing and fusion gene transcription. You would like to keep the oval shape of the spike-ins similar, but just change the GC content, include alleles, or just have minor variations, mutations in very highly similar spike-ins like SNPs. Of course, microRNA is a very hot topic. And a very interesting application would be to have spike-ins of varying integrity, meaning that you got intact full-length spike-ins and you can contrast them to degraded transcripts. And of course, base modification. Now, this is actually also a sort of community effort, so we would like to hear from you what you would like to see as the next module to be realized. If you're interested, just please contact us and send us an email. That would be really, really nice and appreciated. And this is now the last slide, wrapping up actually why you should use the SIRV controls. So you can use the controls to validate your RNA sequencing quantification pipeline. By using the controls, you can control the RNA sequencing experiments, and you can find and research comparable experiments. And this is all done on the transcript level. The nice thing is that actually the SIRVs are platform and sample agnostic, meaning that they are applicable for all sequencing platforms, but also for nanostrings, microarrays, qPCR, and so on. And you can spike them into and combine them with any RNA from any organism or tissue. So with that, I will finish this webinar and say thank you. And I'm now happy to take on any questions. Thanks.
Thank you, Dr. Lukas Paul, for that informative presentation. It's now time for Q&A. If you have any questions that you would like to ask Dr. Paul, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click the send button. We will answer as many questions as we have time for. We have some great questions coming in, so let's get started. Now let's see. Our first question is, Dr. Paul, does the SIRV set include antisense transcripts either partially or completely overlapping the sensed transcript?
Yes, they do. So when designing the transcripts, we have strandedness in mind, and you can look at the gene structure, and we have a number of actually monoexonic, also antisense transcripts, short ones, but also longer overlapping ones, both in the sense and antisense direction. So yes.
Now you mentioned using the 3' quant kit. Wouldn't there not be any or many transcript variants in those sequences? So would the spike-ins help here?
I hope I understand this question correctly. So what you have actually is one read from the 3' end. So this quant kit gets you the sequence from the 3' end of the transcripts, and in the case of the ESCCs, these are unique and you can identify them correctly. When you would use the quant kit to identify the transcript variants of the SIRVs, you would probably fail because there are many of these that share the same 3' UTR sequences, and you could not distinguish these. You could still do gene expression, profiling, and quantification, and that's what this quant kit is actually directed at.
Dr. Paul, are they applicable for small RNA analysis? If not, do you have any spike-in for small RNA?
So the shortest RNA in the SIRVs is actually around 200 nucleotides long. So it doesn't really target now the microRNA size range. But of course, this is sort of in the orbit, in the pipeline, and microRNAs are definitely a module that is also actually heavily discussed at the ERCC 2.0 consortium.
What is the cost of the SIRV Suite software?
So for the SIRV Suite software, actually there are no costs, so it's free of charge. We have installed it in the cloud at Cloudman. You can run a limited experiment there, so all your regular SIRV experiments, you can just upload the data and evaluate it there. If you want to extend it to more experiments, then actually you can clone this instance and install your own Cloudman environment, which of course then would be costing a bit more. However, you can also clone this SIRV environment and get all the tools in a local Galaxy environment on your computer, on your server. So this is totally available, well documented, and for free.
Now, can you store after dilution and use sometime after?
That is a very good question. So I mentioned that we have to ship and store the RNAs at a higher concentration, so a 1,000-fold higher concentration than you would usually use them. And of course, you could say you dilute it 10-fold and then store it at -20 for a week or so. That would be borderline already, I would say. We would recommend to always do a fresh dilution, and that, of course, limits a bit the scope, and that is something that is definitely on the radar, and there should be a formulation where you can have the diluted SIRVs as well.
Dr. Paul, can SIRVs be used for normalization, and if yes, how is this performed?
So the SIRVs are a quality control measurement, meaning that it tells you the biases and also the extent of the biases. You cannot really normalize a particular gene expression based on the values that you get from the SIRVs and also not derive absolute quantification values. There has been a heavy debate about the use of the spiking transcripts for normalization purposes and getting absolute gene expression values. There's one publication where this was performed, but this was for a very defined setting and it was very elaborate, let's put it this way. So at the moment, this is not a standard application of spikings in general.
Is there a way to get SIRV-Suite outside of Galaxy?
So the nice thing is that actually all those tools that I showed are based on command line tools and freely available software. So actually, yes, you could just contact us and we would help you to transfer that also outside of Galaxy.
How many molecules of spike-ins do you add to each sample?
You could look that up in the user guide, actually. I was now talking about the percentage of reads that we want to dedicate to the spike-ins, but of course that relates directly to a certain molarity. And yeah, so that is actually available. So of course we've got quantifications of all our SIRV transcripts in the mixes, and you can derive this value. So for a single cell, you would probably think about 360,000 transcripts that you would get, and if you, for a single cell experiment, would like to spike in the SIRVs, this would be only 3,600 molecules. So we're talking about really low numbers here.
Very good. Now, do we have to use all 3 sets in each sample, or can we use different E-set in different samples like controls and experimental?
That's a really good question, and it addresses actually the different applications of the SIRV. So if you would want to validate your pipeline or look at differential gene expression, then you would spike in different SIRV mixes into different samples. Let's say in sample A you spike in E0, and in sample B you spike in E2. And then you can assess the accuracy of differential gene expression on a transcript level. However, if you then later on want to monitor the quality, the consistency of the quality, it might be enough to actually just use one of the mixes, and then we would recommend actually E0 because already here you see the deviation in the quantification. So it's actually up to the application which mixes you should use.
Have you run the SIRV spiking control data through the ERCC dashboard to generate ERCC dashboard-like metrics, such as limit of detection?
So, yeah, the question was if you have run the SIRV spiking controls through the ERCC dashboard, and actually, no, we haven't. And the thing is that the ERCCs are specifically designed to cover this large dynamic range from 1 to 1 million. We have focused the SIRVs on 1 to 100 range maximum, and therefore a limit of detection is not the target of these controls. And you have to get anyway sufficient reads to be able to detect the transcript variants.
Is the Evaluator available as an open source command line package?
Not yet. Let's put it this way. No, I mean, we've realized it in the Galaxy environment, but I would have to talk to our bioinformaticians how we could do that. If you would drop me an email, I can for sure put you in touch and we can arrange for that.
Is this an open source design?
Definitely, yes. So the whole software and the whole algorithms behind this SIRV-Suite are completely open.
What is the difference between Lexogen RNA controls and the ERCC spike-in? So the ERCC spike-ins are designed as monoexonic non-isoform controls, meaning all of those 92 sequences are unique, and that makes it actually quite easy downstream to assign the reads to the correct ERCCs.
With the SIRVs, it's exactly the other way around. We do not have that many genes, only 7, not 92, but we have about the same number of transcripts that map to them, and therefore we've got a much higher complexity, and the whole pipeline, and especially the software part, must be really very capable of assigning the reads to the correct transcript, because only when the reads have been assigned correctly, you can quantify the transcript and the gene accurately.
Okay, let's see. We have so many great questions coming in. Have you used SIRVs to benchmark transcript assembly tools? If so, what did you find?
So actually, we ourselves have not done that, but I cannot exclude that many of our SIRV customers or test partners have done that already, and it would be a really, really good application for the SIRVs.
And I believe we have time for one more question. What is the fraction of transcripts that are greater than or equal to 1 kb?
So it must be actually more than 50%, because the median is 1.1 kb. So we are talking about 35 to 40 of those 69 transcripts that are longer than 1 kb.
Dr. Lukas, I would like to thank you once again for your presentation. Dr. Lukas Paul, do you have any final comments for us?
I just would like to thank you especially and the whole organization at LabRoots for enabling this webinar. It was my first one and I really enjoyed it, and thanks for all these questions, and I really hope that you will be in contact with us, and please visit our website, get known to the SIRV suite, and contact us. Thank you.
Thank you again, Dr. Lukas Paul. And I would also like to thank our sponsor, Lexogen, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through April 19th, 2017. You will receive an email from LabRoots letting you know when this webcast is available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us. We hope to see you again soon. Goodbye.